Here's a real-life example of how PTMScan can facilitate translational discovery. Non-small cell lung cancer, NSCLC, has a very poor prognosis, particularly when it isn't caught until the later stages. Historically, there have not been many treatment options available to patients. Knowing that many cancers are driven by an excess of tyrosine kinase activity, CST used a PTMScan phosphotyrosine antibody to enrich for phosphorylated peptides from 41 non-small cell lung cancer or NSCLC cell lines and 150 NSCLC tumors. Normally finding the right markers is like looking for a needle in a haystack. But using PTMScan is like holding a finely tuned magnet above the stack, enriching the sample and making it easier to find what you're looking for. This analysis revealed that the tyrosine kinase ALK was present and activated in a subset of the samples. Deeper investigation, in fact, revealed novel fusions of EML4 and ALK. Additional studies indicated that this fusion protein is highly oncogenic, and 3.7% of NSCLC patients express it in their tumors. In other words, we found our needle. Studies also showed that cancer cells expressing the EML4 ALK fusion protein are sensitive to the small molecule ALK inhibitor crizotinib, which the FDA approved in 2011 for the treatment of ALK fusion protein positive NSCLC. Instead of stopping there, CST developed a highly specific and sensitive ALK antibody that detects full-length ALK and the EML4 ALK fusion protein. The FDA approved an immunohistochemistry (IHC) companion diagnostic test that uses the ALK D5F3 clone licensed from CST. This will help physicians determine which NSCLC patients may be helped by crizotinib treatment, giving patients who once had few options a new ray of hope. So what's the real value here? CST not only identified EML4 ALK as a major driver of NSCLC, which can respond to an FDA-approved drug, but went a step further and developed an antibody that can be used to test for which patients could be candidates for treatment. All thanks to the power of simplified proteomics. To find out more about how PTMScan can simplify your proteomics work and change the way you think about discovery, click here.